Welcome to our video lecture on topic A2.1, Origins of Cells. Our guiding questions for today: what plausible hypotheses could account for the origin of life, and what intermediate stages could there have been between non-living matter and the first living cells? Our objectives: we're going to describe conditions on early Earth as we think they were. We're going to review cell theory and the functions of living things. There are 7 of them. We're going to describe investigations that have led to our understanding of abiogenesis, the forming of life from things that were not living. We're going to describe some of the stages of abiogenesis. We're going to define this thing called LUCA, the last universal common ancestor. We're going to outline some of the evidence that we have for the existence of LUCA, and then we're going to talk about how sometimes it's really hard for us to figure out all this stuff. What we know, what we think we know about conditions on early Earth, definitely lots of indirect information. No one was around 5 billion years ago to make any observations, record any data. And so instead we look at lots of indirect information. We look at other parts of the universe and make guesses as to what our solar system looked like a long time ago. We look at rocks, we look at the chemical compositions of different ages of rocks, and that helps us to make some guesses about what stuff used to be like here on Earth. What we are pretty sure we know, and again, we have these giant paradigm shifts in science, and so there might be some better technology at some point in the near future that allows us to make better observations, better predictions, and so this might change, but for now, our best theory about the beginnings of the universe was this Big Bang theory. So we're going to use G for giga. Giga is in the metric system 10 to the 9 for us here in the United States. That's a billion. So 13.8 billion years ago, the Big Bang happened. There was a chunk of matter and it exploded and it moved away from the other pieces of matter. The universe is expanding. We can observe our universe expanding. And so that's some of the evidence that we have for Big Bang. About 4.6, again, G for giga or billion years ago, the sun began to form. Gas and dust and other stuff was attracted to each other due to gravity, gravitational forces, and though we ended up with this mass, lots of hydrogen, and the hydrogen was squished into helium, and then that nuclear fusion, we had lots of energy, so lots and lots of energy coming out of the sun. Actually, not as much as we have now, interestingly. So the sun is definitely ramped up a little bit. More efficient energy production than it used to be. 4.5 billion years ago, so just shortly after the sun formed, the Earth began to do the same thing. Again, lots of gas and dust and gravitational forces merging all these pieces together, and we ended up with this early Earth. Earth had a lot of volcanoes. There wasn't much of an atmosphere, and so all kinds of things were smashing into the surface of the Earth, so many meteorites. Because we didn't have an atmosphere, it was super, super hot. We didn't have any oxygen in the atmosphere, so UV radiation was not blocked by the ozone layer. Lots and lots of lightning, so much energy, crazy high temperatures, and eventually over time we did start to accumulate some gases, some of the gases that we think were in this early atmosphere, oodles of carbon dioxide, lots of methane. CH4 is methane. Probably lots of sulfites as well, so sulfur-containing compounds, since we have all those volcanoes. Not a lot of oxygen in the air because a couple reasons. All of this hot rock is going to react with the oxygen, and so that oxygen would form oxides, iron oxides, especially given all that hot rock, all that magma and lava. Again, because there wasn't any oxygen, there wasn't any ozone layer, so lots of UV radiation, making the surface super energetic, super hot. Lightning, again, a big source of energy. So early Earth, crazy warm, crazy energetic. In 1929, a scientist by the name of Haldane proposed this idea of a hot, dilute soup, the primordial soup, here on Earth. And the idea was because of all the volcanoes and all the meteorites and all the energy and the lightning and whatnot, that we would have a lot of dissolved particles, molecules in the water on the surface of the Earth, this hot, dilute soup. We would also have some hydrothermal vents in the oceans, hot springs on land, providing more energy in the form of heat. All these dissolved particles would then run into each other. Remember kinetic molecular theory, we know that molecules, when they collide with sufficient energy, can carry out chemical reactions. So we're going to have lots of stuff in this primordial soup, and it's just going to be bouncing around, running into each other, making some chemical bonds. Lots of chemical reactions are happening. Carbon, because remember that carbon has its 6 protons, it's got 2 electrons in the first energy level, and then 4 valence electrons, 4 electrons in the outermost energy level, that valence energy level. This means that carbon has so many ways to make lots of bonds. And so carbon and other mini compounds of carbon floating around in the primordial soup are going to be able to make all kinds of super cool reactions, lots of compounds formed. One of the other things that carbon does really well is catenation, where we can start to build chains of carbon compounds, carbon pieces to make polymers, polymer, big long chains made of littler pieces of molecules. We know that we also have lots of meteorites and comets that would bring more pieces and parts, more molecules, more substances to add to this primordial soup. We actually have found on meteorites amino acids. Amino acids are the pieces and parts that make up proteins. How cool is that? We have baby proteins on meteorites in space. It's amazing. So we've got these compounds, and then they react with each other to make bigger compounds, and those compounds begin to go through some catenation. We've got some organic chemistry happening, and then we end up with some super complex molecules and polymers. These polymers begin to get wrapped up in some vesicles. Those vesicles start to replicate themselves. They begin to specialize, and we end up with cells, and we have a life, and it's amazing, amazing. And then, of course, those cells are going to change the environment. And you might be thinking, wait a minute, how in the world do we get from carbon dioxide and methane to whole entire cells with mitochondria and ribosomes and all this stuff? We're going to talk a lot about those pieces and parts, those stages. But I do want to say that we can do some of this in lab, not all of the stages, but in labs we can do some of these pieces. For example, at the University of Georgia, fairly recently, in the last decade or so, they were actually able to take cyanide, which is just some carbon and some nitrogen, and they were actually able to form adenine, which is one of the nucleotides that makes up DNA. So if you're like, wait, how do we get from carbon dioxide all the way to DNA? We can actually kind of do it little pieces and parts, even in the lab today. So cyanide can turn into adenine with a lot of help, but it can be done. That's amazing. We're going to talk about cells, the arising of cells here on Earth. Let's review real quick cell theory. So these first 3 pieces are the most important, then we throw these other ones in here because they are also relevant. But if I ask you to write down the tenets of cell theory, I'm really looking for those first 3. All living things are composed of cells. Cells are the basic units of life. Cells come from pre-existing cells. We also know that single-celled organisms, so you can be one cell and still carry out all 7 functions of life. Multicellular organisms like us will have specialized cells. Let's remind ourselves, what are those functions of life? Here they are. Homeostasis, maintaining those conditions necessary to stay alive. Metabolism, the sum of chemical reactions that are usually enzyme catalyzed. We'll talk a lot about enzymes when we get to our unit on metabolism. Nutrition, getting food. Excretion, getting rid of waste products. Growth, either our cells growing bigger or us making new cells. Response to stimuli. Sometimes we need to adapt to our environment, and our ability to respond and stay alive in our environment is that response. And then, of course, reproduction. How do we keep our species, our cell lines alive over time? This is huge. Cells come from pre-existing cells. But where did the first cell come from? Cells only come from pre-existing cells. Cells haven't always existed. So at some point, we had to have a cell that didn't come from a pre-existing cell. We're going to talk about that now. A long time ago, a couple hundred years ago, we thought of this idea. Spontaneous generation was how we could explain life, literally spontaneously generating. People would leave meat out, the meat would rot, and then all of a sudden there would be maggots on the meat, and sometimes there would be rats. There would be no rats the night before, but in the morning there are rats on the meat. So clearly the maggots in the rats must have just popped out of the meat like poof, magic. Spontaneous generation. We literally thought that living things could just spontaneously appear on rotten meat. And then this guy, Louis Pasteur, came along and he was like, that doesn't make any sense. So he did this investigation with what we call swan neck flasks. These are his swan neck flasks. So he put some yummy, juicy, brothy stuff in here. Lots of things that living things would like to eat. Bacteria and mold, at least, would like to eat, like dog pee and stuff like that. And he boiled it. He boiled it, killing all the microbes in the broth. But he didn't know that microbes existed yet because we didn't have microscopes powerful enough to see bacteria. But he was like, let me see what happens if I heat up this broth of dog pee and other stuff. So then he just left it for a really long time, a really, really, really long time. Nothing grew in the flask. The flask stayed sterile. So he tried again, boiled the broth, which we know now, killed the microorganisms, but then he broke the neck off. Didn't take very long at all for microorganisms, mold, and bacteria to start to grow in the broth. So Pasteur was like, if spontaneous generation were a thing, we would have seen that growth in this flask. But since we only saw it in this, clearly something had to fall into the flask in order for that life to arise. Another guy, Robert Remick, looked at frog cells, frog zygotes. And so he looked at an egg that had been fertilized and he watched it literally divide into two. And he was like, whoa, that's amazing. So all of these cells had to come from another cell. And so he also was able to provide some evidence that cells come from pre-existing cells and not that they pop up out of nowhere. But before we can get cells, we need some molecules. In 1953, Stanley Miller, a student at the University of Chicago, and Harold Yuri, his professor, designed this investigation in which they were trying to model Earth's early environment. So they had some water and they heated the water kind of like the oceans were super hot. And so we had this water vapor. They added into this flask, we had some ammonia gas and we had some methane and we had some hydrogen gas. And so the water vapor comes and mixes with the ammonia and the methane and the hydrogen gas. They also added some electrodes to simulate lightning and the UV radiation that would be coming through to the surface of the Earth. And they just ran this thing for like a week. And after a week, the stuff down here started to turn like pink and then red and then like this dark brown. It was amazing. And so they took out some of the solution, some of the samples, and discovered more than 20 different amino acids. Amino acids are the building blocks of proteins. More than 20 different amino acids had been produced over the course of that week. Lots of other organic molecules were also in there, but those amino acids were huge. That was exciting. One of the challenges, though, is do we even know that these were the gases that were present in the early atmosphere? Do we even know how much water vapor there was? Do we know the temperature of all that stuff? Do we know how much lightning there was? So this model was a good one, but it definitely had some limitations. So over time, since 1953, lots of other scientists have been carrying out similar investigations, slightly different concentrations, different gas sources, and just about every time we're still pretty successful in making some organic compounds. Super cool, amazing. Lots of evidence that the primordial soup, that early atmosphere, early conditions on Earth could lead to the production of at least some pretty big organic molecules. If this sounds super interesting to you and you want to know more, highly recommend on YouTube the Stated Clearly series, What Was the Miller-Urey Experiment? They have really, really nice explanations. So we're feeling pretty confident that we could on early Earth make some of those organic molecules. And we could go through some of those polymerization chemical reactions. Because it was hot and energetic, we could make some big organic molecules, that polymerization piece. But going from big organic molecules to cell membrane and then actual cells, like those are some pretty giant steps. So let's talk about this origin of vesicles, these protocells, progenotes, sometimes we call them. How did we get these organic molecules wrapped up in vesicles so that we could have this maintenance of internal conditions, homeostasis, start to occur? The answer is phospholipids. So phospholipids, the molecules that make up our membranes, are amphipathic. That prefix, amphi, means both, and path means feel. These guys feel both polar and nonpolar. They are both hydrophilic and hydrophobic. And so these phospholipids, when they're in water, they will form spontaneously all by themselves without any direction from a scientist or us. Well, we are scientists, so I guess that was a redundant statement. They'll form these little bubbles called micelles. Micelles, soap does this also, so don't go to the store and buy the fancy bottle of micellar solution to wash your face, because it's pretty much just soap. So don't go crazy with the micelles. Anyhow, so these bubbles, these soap bubbles, will spontaneously form because phospholipids are amphipathic. If we have some kind of polar organic molecule, like a little piece of RNA or maybe some amino acids, then the phosphate heads that are polar will be attracted to that polar chunk of amino acid or whatever we have, and we'll end up with a phospholipid bilayer where the polar heads are attracted to the polar molecules. These polar heads are super happy to sit out in the water, and then all of our non-polar fatty acid tails are going to stick in toward each other. Crazy cool. These protocells, not quite cells, but these bubbles, membrane-bound bubbles of amino acids or protein, can actually spontaneously form. How cool is that? So we have organic molecules, we can polymerize them to make them big organic molecules, we can bubble them up in vesicles, phospholipid bilayers, so that we can maintain some kind of internal condition. But there isn't any way for us to reproduce these bubbles, these not yet cells, because they don't have any genetic material. This is a super important piece of being alive, reproduction. And you cannot reproduce, you cannot pass your traits on to offspring without some kind of genetic material. DNA is in cells. That DNA codes for proteins. The proteins can sometimes be enzymes. Those enzymes carry out metabolic processes. One of the things that enzymes can do is actually copy the DNA. So you have to have enzymes to copy the DNA, but you have to have the DNA to code for the enzyme to copy the DNA. So what came first? The DNA or the enzyme? We actually think it was neither. We think it was RNA. Scientists have proposed the RNA world hypothesis, which states that we think RNA came before DNA. And some of the reasons for this are RNA can store genetic material just like DNA can. RNA is self-replicating. It has what we call base pairs, adenine and uracil, cytosine and guanine. And these guys can kind of pair up with each other so that we can have copies of the sequences of RNA. And what makes RNA super way cooler than DNA is that it's catalytic. It can actually help make chemical reactions happen. It is a catalyst itself. There are some viruses like this guy. This is a retrovirus, and this virus is not a living thing, but it is something that affects us living things. And it does have some RNA instead of DNA. It has an enzyme called reverse transcriptase, and then once the RNA and this enzyme gets into a host cell, that reverse transcriptase can actually copy the RNA back into DNA, and then there are lots of things that we'll talk about in another lecture. But there are viruses out there that are RNA based. We also know that there are some chunks of RNA that can act like enzymes, and we call those ribozymes. And here are just a couple different examples of ribozymes. These guys can help make chemical reactions happen more quickly, more efficiently, with less energy required. Ribozymes can help make chemical reactions happen. Ribozymes are also parts of ribosomes. So here is a piece of a ribosome. This is the large subunit. And ribosomes are composed of a couple different things. The orange and yellow pieces, that's RNA. And then the blue pieces, or purple, I guess, is protein. So ribosomes are composed of protein and RNA. What's crazy cool is this ribosome with its RNA can actually cause chemical reactions to occur. This is how we build proteins. This little green spot here in the middle is the active site. This is actually the piece of the ribosome that forms the bond between the amino acids to make the proteins. It's amazing. So RNA could have been an early genetic information storage molecule because it's not quite as fussy as DNA. It can do a lot of stuff on its own, whereas DNA needs a whole lot of support from lots and lots of enzymes. To recap, we had organic molecules in primordial soup with lots of energy that could make bigger organic molecules. And then we have phospholipids that can build vesicles around them, and we've got RNA, potential genetic material, so that these things can pass on traits to their offspring. My friends, I think we have living things. This idea of going from non-living material to living material, going from not life to life, is abiogenesis. So making living things from not living things, abiogenesis. Crazy cool. We know, of course, that living things have changed a lot over time, and we actually have some names for those things that used to exist a really, really, really long time ago. We have LUCA, the last universal common ancestor, So basically the cells that all of the living things that are still on Earth today have evolved from. There were definitely other ancient cells that had descendants, but those guys have all gone extinct. So we're not super concerned about these guys over here. We're really just looking at the last universal common ancestor, looking at all the things that are still on planet Earth today. What do they all have in common? From what did they all evolve? And LUCA is it. Before LUCA was FUCA, which sounds slightly inappropriate, I feel like. This was the first universal common ancestor, which gave rise to all the cells, even the ones that are over here that have gone extinct and are not part of our inheritance lineage. We have lots of evidence for the existence of LUCA. One of those things is the Universal Genetic Code. We'll talk about that on the next slide. We have lots of conserved genes. Conserved means saved. There are genes, genetic information that is the same, have been saved over time and exist in all of us, things that are still alive on Earth today. If we all have them, they probably had to come from our common ancestor. One of the other things that's been saved over time is ribosomal structure. Our ribosomes, not exactly the same, but the ribosomes of bacteria and archaea and eukaryota, all of our ribosomes are pretty similar in terms of structure and function, which is crazy cool. Polymerases are enzymes that help to copy, produce RNAs and DNAs, also really similar from one organism to the next. All of these things give evidence to the existence of a common ancestor. And the last one for everything that existed on Earth today is the last universal common ancestor. I mentioned this idea of a universal genetic code. Basically, all of us living things on Earth use the same decoder ring. Okay, or codon chart is probably better. Do we even still have decoder rings? I'm not sure. That might be an old person thing. Anyhow, codon chart. So codon charts help us determine, looking at a messenger RNA sequence, what the amino acid sequence in a protein is going to be. So a codon is a triplet of 3 messenger RNA nucleotides. Remember that we have A and U and C and G, adenine and uracil, cytosine and guanine. So with very, very small variations, most of us from E. coli up to IB biology students are going to be using the same code. So if I have a messenger RNA codon and my first nucleotide is uracil, and my second nucleotide is guanine, and my third nucleotide is cytosine, then I'm going to end up with the amino acid cysteine in my protein, because me and the E. coli and a banana are all going to use pretty much the same code. crazy cool. It's really hard for us to figure out how long ago Luca, the last universal common ancestor, existed, because cells, little things, squishy stuff, do not fossilize very well. We do have some super amazing fossils. These guys, these trilobites, like 545 million years old. That is crazy to think that we can look at rocks of living things that were here that long ago. But stuff like Luca is not going to be preserved in a fossil record. We do have some really old fossils. These rocks are called stromatolites. They are fossilized chunks of cyanobacteria. Some bacteria are photosynthetic. Cyanobacteria are one of our strains of bacteria that can photosynthesize. And sometimes they get stuck together. And these guys are rocks, fossilized blobs of cyanobacteria. And by looking at different ratios of radioactive carbon isotopes, carbon 14, carbon 13, carbon 12, ooh, that was supposed to be a 13. By looking at the ratio of these different radioisotopes in a sample, we can make some predictions about how old they are. And we think that some of these stromatolites that we find in Australia could be like 3.4, remember that G is for giga, billion, 10 to the 9 years old. That's crazy. But we know that life probably existed before cyanobacteria, because where did the cyanobacteria come from? Where did Luca come from? One of the things that we have now, which is a crazy, amazing, big brain idea, are these molecular clocks, or sometimes we call them genomic clocks. Woo, if I could spell genomic clocks. So basically the idea is this: we look at DNA sequences changing. We think it's like once every 40 copies of DNA we get a mutation. And so we can look at this rate: how fast does DNA mutate, how quickly do changes in sequences appear. And then we can use that rate of changes in the sequences appearing, and we can look at how many differences in sequences there are between two things, and then we can figure out how many million or billion years ago did those species diverge. So we can use this kind of clock, this rate of genetic change, look at how many differences there are in the genes of different species, and we can figure out that stuff was really old. And we think that the first living things on Earth probably dated back to about 4.5 billion years ago, 4.5 billion years ago, which is about the same time that this planet called Theia collided with early Earth, and that's how we got the moon. Can you believe that? Crazy. So amazing. We used differences in the DNA to try to figure out how old Luca is using that genomic clock or molecular clock. We can also look at the similarities in genes of all living things that are still on Earth today to make some assumptions about what genes Luca must have had. If bacteria and archaebacteria and also us eukaryotes, if we have a lot of the same genes, then we can assume that those must have come from our common ancestor. They must have also been genes that Luca had. If we have them all the same, then probably Luca had them too. That's probably where we all got them from. There are just about 355 families of proteins that just about all living things still can make. We don't always make them, but we have the genes in order to make them. And we are working on the assumption that Luca probably evolved near hydrothermal vents, because these protein families, these genes, are the genes that are necessary to survive near hydrothermal vents. We have genes, proteins for anaerobic respiration. We'll talk about that more in our unit on respiration and metabolism. The fixation of carbon dioxide and nitrogen, all of these genes are necessary for survival in conditions like those near hydrothermal vents. So probably Luca must have evolved near a hydrothermal vent. This is where life started. Our last universal common ancestor near a hydrothermal vent. And just in case you were wondering, what even is a hydrothermal vent? Well, let's look at some geology here really fast. So here is the crust of the Earth under the ocean. Here's our ocean crust. And there's magma, that molten rock that is under the crust. And sometimes it bubbles up a little bit. And where it bubbles up under the ocean can form these hydrothermal vents. So it's water, but it's also super hot because it's magma. It's molten rock. So we have lots of hot, and then it gets a little bit less hot because it's like super deep in the ocean. And so because of the hot and the less hot, we have lots of swirly things happening. So lots of convection currents. We have lots of minerals coming up from the magma. So we have some sulfides. This is hydrosulfuric acid, carbon dioxide, and methane, and some manganese, and some iron, and some helium, and all kinds of crazy things. Lots and lots of chemicals, lots of nutrients, lots of energy here near these hydrothermal vents. And so this energy, these nutrients, these resources available probably is where Luca, our last universal common ancestor, existed. And just a reminder, we have been working in our current paradigm. So we've been collecting some information, and we're using that information to put together puzzle pieces, and we're like, okay, this makes sense. But do not be surprised if at some point with some better piece of technology, we are able to figure out some stuff that doesn't quite fit in our current paradigm. And then we're going to have to start to shift our idea of how all these pieces fit together. And so right now, Big Bang and Luca, these make sense given the information that we have, but it wouldn't surprise me at all if there isn't a big paradigm shift in your lifetime, because we'll have more information, better technology. We can come up with even better explanations for how life has evolved here on Earth. Good work, my friends. We have come to the end. Guiding questions reviewed. Remember that we talked about plausible hypotheses that could account for the origin of life and the intermediate stages between non-living matter and those first living cells. We talked about the organic molecules that we could make bigger with polymerization. We looked at vesicles, and then we talked about RNA as the first potential genetic material. Our objectives, we described those conditions on early Earth, the ones that we think were there. We reviewed cell theory, the functions of living things. We described investigations that lead to our understanding of abiogenesis. How did cells arise on Earth? We talked about those stages, again, kind of those same stages that were here. We defined Luca, our last universal common ancestor. We talked about some of the evidence that we have for Luca, and then we talked about this idea of a paradigm shift, and sometimes the models that we have now, the assumptions that we make now aren't the best. And once we get some new information, some new technology, we can adjust those ideas and have a brand new paradigm. Good work today.